Hello everyone, so this is the GCSE Combined Science Trilogy Biology Paper 2 that was sat by pupils in 2018, and this is the higher tier paper. I'm going to go through and demonstrate how I would have tackled this paper. I've put a link to the blank paper and the mark scheme in the description, along with revision videos, question by question, that link to the specific content if you need to go back and revise it. So, getting started then. Question number 1. Many biotic and abiotic factors can affect the growth of plants. Are the factors in Table 1 biotic or abiotic? Well, biotic are all to do with living factors, and abiotic are non-living factors. So we just need to identify from Table 1, is it a biotic or abiotic factor? Well, diseases are caused by pathogens, bacteria for instance, so we would say that is a biotic factor. Herbivores, animals that eat plants, well that would be biotic because they are living. Whereas temperature and water, these are not living features or organisms, so therefore it would be abiotic. 2 students investigated the effect of light intensity on the distribution of small plants. The plants are growing under a tree in a park. The students made the following hypothesis. As you move outwards from a tree, there will be more plant growth. Explain why the students thought their hypothesis would be correct. Well, this is now linking back to your knowledge of plants. Well, we know that if you move outwards from a tree, there would therefore be more light. What process is light important for? Well, it would be photosynthesis. And then we can link it to what can photosynthesis, what does it produce? Well, we know it makes glucose, which can then be used to help the plants make protein for growth. So we could structure the answer like this. As you move out from the plant or tree, there will be more light. More light means more photosynthesis. As you have more photosynthesis, you'll have more sugar and more glucose produced, which can also be used and converted to protein to increase plant growth. So therefore, more sunlight, more plant growth, and that's why they thought their hypothesis would be correct. Question 1.3 then. The students used 2 pieces of equipment. Give the scientific name of each piece of equipment. So a square frame measuring 0.5 metre by 0.5 metre and an electronic device to measure light intensity. Well, hopefully you went onto the field when you were at school and maybe you were counting daisies or a different species, and the metal grid you used would be a quadrat. Now, quadrats don't have to be half a metre by half a metre. Quite often they're 1 metre by 1 metre, but they come in different sizes. An electronic device to measure light intensity. Now this question threw a lot of people. Well, it's quite a simple answer. It's just a light meter. This is the method the students used. They fix one end of a tape measure to the base of a tree. They fix the other end of the tape measure 11 metres from the tree. At 0 metres, they put the square frame on the ground. They identified all the plant species growing inside the frame. They estimated and recorded the percentage cover of each plant species. They measured the light intensity inside the frame. They put the square frame on the ground every 2 metres along the tape to 10 metres. And they repeated steps 4 to 6 in every frame. So what they're effectively doing is moving their metal grid 2 metres along each time. They're putting it down, they're recording the light intensity, they are then recording the amount of organisms and plants in the quadrat at that point. So the question is, calculate the total area sampled, and it's only 1 mark. So we don't overthink it. Well, we've got our quadrat and we know it's 1/2 metre by 1/2 metre. So if we do 0.5 times by 0.5, that gives us 0.25 metres squared. That is the area of 1 quadrat. We then got to know how many times they put it down. Well, we can go through that. They would put it down another time here, then another 2 metres along, another 2 metres, another 2 metres, another 2 metres. So including our starting quadrat, they would have put it down 1, 2, 3, 4, 5, 6 times. 6 times 0.25 gives you a grand total of 1.5 metres squared. Question 1.5. The whole investigation was done as quickly as possible on the same day. Suggest a reason why. So you want to think about what is the main control variable for this experiment. Obviously, as we discussed in the previous question, it's all to do with light. So why was it done so quickly? Well, we want the light intensity to be constant or the same. Otherwise, the results could be affected. Any other references to temperature, weather, sun isn't specific enough. You've got to say light intensity. Give one way the investigation could be improved. Well, in this investigation, if you could look back at the method, they've only done it once. Okay, so you can repeat it using different transects. You could do it again under different trees or at different areas. They did it every 2 metres, where you could sample every 1 metre. So in questions like this, just go back to the method and see if you can think about anything that you could change to increase the accuracy or the repeatability of the experiment. So question 1.7 then. You've been given a table of their results. Which species in table 2 will only grow at the high light intensity? So we know as you move away from the tree that the light intensity is going up. So we can know as we go up, you have a higher light intensity. Which species, okay, is only growing at the high light intensity? Well, we can see it is daisies. Hopefully you would have seen this when you went onto the school field and counted the amount of daisies. You would have seen them a much higher percentage cover in areas where there is more sun. Question 1.8. What conclusion can be made about the relationship between light intensity and the total percentage cover of plants? Use data from table 2 in your answer. So if we look back at the table, you can see that generally, as light intensity goes up, you're getting more and more plants. Okay, in terms of the total cover, it goes up and up and up till about 4 metres, and then it hits 100%, and then it stays at 100%. So this is a 2-mark describe question, and it's specifically looking at this row here. So we need to describe the pattern that is shown. If we were going to draw it on a little sketch graph, it would look a bit like this. You would have your light intensity as your independent variable, your percentage cover as your dependent variable. It would go up and then it would level off. So this is what we need to describe. So what conclusion can you make? You're linking in data where you could say, as light intensity increases, as does plant cover. Now that would be enough to get you 1 mark, but then we need to link in some data. So we can say the point at which it flattens off. And if we look back at the data, well, it reaches 100% at 6 metres. It reaches 100% and levels off. So we've described the pattern in the table and we've used some data from it to support the answer. Question 1.9. Light intensity might not be the cause of this pattern of plant distribution. Suggest one other factor that may cause these results. Give one reason for your answer. So now you've got to think about any other factors that may affect the level of plants apart from light intensity. Well, carbon dioxide is another limiting factor of photosynthesis, but that would be the same in the atmosphere, regardless of where it is, so it can't be that. So what other factors may affect the amount of plants? Well, it could be the amount of water or rain at different locations, okay? And that might be because if you've got increasing tree coverage, the leaves are stopping water reaching the ground, or the trees themselves are absorbing it. Therefore, there's no water left for the daisies or other plants, and therefore it's not increasing. That might not be the only reasons though. You could also say, well, maybe there is a lack of mineral ions in the soil. Okay, again, maybe because the trees have absorbed it. Now, I think those two would be your go-to answers for this question, but you could also say that because there are so many leaves, the temperature isn't as high because it's blocking out the thermal energy, therefore there wouldn't be as many plants growing. Okay, so question number 2 then. Pseudomonas bacteria causes infections in hospital patients. A new strain of bacteria has evolved. The new strain can only be killed by one type of antibiotic called fluoroquinolone. Scientists want to prevent the new strain of Pseudomonas from spreading in the human population. Explain the advice that doctors should be given to prevent the spread of the new strain. So we really just want to focus on what doctors can do to prevent the overprescription of this antibiotic. Because if a pathogen is exposed to the antibiotic more and more, there's a greater chance of them mutating and becoming resistant to it. So what should doctors do to prevent or reduce the amount that they prescribe it? Well, it can all come down to, first of all, the severity of illness. They must only prescribe if it is severe illness. If not, let the patient's immune system deal with it. Point 2, the doctor would then have to be sure that actually the drug they are prescribing is going to work. So they must not prescribe it for viral infections. Because we know, because viruses exist inside our own body cells, that antibiotics can't get to them and they can't kill it. Because antibiotics do not kill viruses. So you can see what we're doing here in terms of the answer. We're making a statement about what they must not do, and then straight after we're putting a reason or a suggestion why. The final one that we can say is that if the doctor does prescribe it, the patient must finish the course because there's a greater chance of the bacteria mutating if the pathogen is allowed to be exposed to the antibiotic but is then not fully killed by it. To stop the pathogen or the bacteria mutating. Okay, So that would be enough to get you 6 marks on that question. Remember, point, then explain. Point, then explain. Question number 3 then. Scientists have removed microorganisms from inside rocks in the caves in Mexico. The microorganisms have been trapped there between 10,000 and 50,000 years. The caves are very hot, humid, and acidic. Why are these microorganisms called extremophiles? Tick 2 boxes. Well, an extremophile is an organism that lives, quite simply, in extreme habitats. Okay, so let's have a look at the reasons, and we're thinking, how do they link to this definition? Is it because they are thousands of years old? Not quite, no, that's nothing to do with their habitat. They survive in high humidity. Well, that's to do with water levels in the atmosphere, possibly. Let's have a look. They survive in very high temperatures. So because it is hot, that would be classified as an extreme environment, like the desert. So it's likely to be that. We can put 1 tick. They survive in the dark. Well, possibly, but let's see what other options there are. They survive inside rocks. Probably not. They survive where it is acidic. Now, acidic, which one is kind of more extreme? Acidic, or having loads of water, or being in the dark? I would personally go with acidic, because living in an acidic environment, only a very few organisms are adapted to do that. There are many more that live in high amounts of water and inside the dark. So your 2 options would be high temperatures and high acidity. The microorganisms have been inactive for thousands of years, but the scientists have reactivated them. Figure 2 shows the results of enzyme analysis on 4 enzymes, A, B, C, and D. 3 of the enzymes from the microorganisms were found in soil near the caves. 1 of the enzymes was reactivated from microorganisms from the cave. Which enzyme comes from the microorganism from the cave? Okay, we've got our enzyme graphs here showing it how they perform in temperature and pH. So for this question, we're going to have to look for a hint. Now, I can't see any clear hint at the top part of this question, what might be the answer, so we can always go back. Good exam technique, the questions always flow, so look where there might be a hint. And we've already looked at it and we've seen that they survive in hot, humid, and acidic conditions. Now, we've been given graphs relating to temperature and acidity. So which one survives in really high temperatures? Well, that's C. And which one survives in really acidic conditions? Well, on your pH scale, remember that the lower numbers, 1, 2, and 3, are your acidic, your strong acids. And again, that one is C. So we can say that the one likely to be from the cave is C. Give reasons from your answer from 3.2. Well, we've just said it. It has a high optimum pH in acidic conditions. And it has a high activity at high temperatures. Okay, now it's always probably good in these conditions just to quote some data. So pH 1 to 3 and high temperature about 65 degrees Celsius, just to support your evidence. Question 3.4 then. Carl Woese developed the 3-domain system of classification. Describe his 3-domain system. So remember, with classification, there are 2 systems. There was the Carl Linnaeus version, which is the kingdom, phylum, class, order, genus, species, the big rank order with the 5 different kingdoms. And then you've got the more recent one, which was developed by Carl Woese. And his one is based on the 3-domain system, and it's based on DNA analysis. Okay, and he basically said there are 3 domains instead of the 5 kingdoms. The first one is eukaryotes, okay, animal and plant cells, because they have a nucleus. You've then got the prokaryotes. These are ones without the nucleus for your bacteria. And then the final group, these are your really, really simple bacteria, which are called the archaea. Okay, simple, primitive bacteria. Question 3.5 then. Most of the microorganisms from the cave were classified as belonging to the archaea domain of the 3-domain system. Explain why. Well, it's because most archaea, most simple, primitive bacteria, are extremophiles. They are extremophiles. They live in extreme conditions. And that is why they're generally classified as being quite simple, because they haven't evolved, because they've been in very, very extreme conditions. Question 4 then is talking about the nervous system, and it shows a reflex arc with our different neurons in it. Name parts P and Q. Well, P is pointing towards the gap between 2 neurons, so the gap is called a synapse. Q is a connecting neuron that might be in the spinal cord, and that type of neuron that connects the sensory neurons, which carry the information to it, and the motor neurons that carry it to the effector. These are called the relay neurons. Compare how the information is transferred along a neuron with how information is transferred across gap P. So, how is information sent along a nerve cell compared to being how it is transmitted across a synapse? Well, along a neuron, we should know that neurons conduct messages via electrical impulses, whereas across a synapse, this is where, at the end of a synapse, okay, and this is the next part of the neuron, you have neurotransmitter, or a chemical that diffuses across the synapse. Okay, so a synapse is the movement of chemicals, and it'd be really good if you can throw in the keyword neurotransmitter. Question 4.3. Why does a conscious action take longer than a reflex action? Well, the conscious action is determined by the brain, whereas a reflex action is controlled by the spinal cord. So why does a conscious action take longer? Well, quite simply, it has potentially further to travel. So the signals that are sent around the body have further to travel, and also the brain has to think and decide on a course of action. The spinal cord doesn't think, it just sends a message right back about what to do. Question 4.4 then. Information travels at 120 meters per second in neurons. Calculate the time it would take for the information to travel 1.6 meters along a neuron. Give your answer in milliseconds. So, this is a maths question in a biology paper. There's no specific formulas you need to remember for biology paper 2, so it's going to be a bit of a logic puzzle. Now we've been given, look at what variables you've got. You've got meters per second, which is speed. You're asked to calculate the time, and you've been given distance. So hopefully you remember the classic equation that you should have done since key stage 3. Speed equals distance divided by time. They're asking you to calculate the time and they've given you speed and distance. So I can rearrange this equation or put it into the speed triangle. If speed is distance over time, time is distance divided by speed. So I can then substitute in the numbers. So it'd be 1.6 divided by 120 meters per second. And that would give me an answer of 0.013, okay? But that would be in seconds because it was meters per second. We want it in milliseconds. Now what we can do then, to turn it into milliseconds, you're going down to a smaller unit, so you times it by 1,000. 1,000 milliseconds in a second, so therefore it should be 13.33 milliseconds. It doesn't say about decimal places or significant figures, so as long as you put in 1 or 2 decimal places, that's absolutely fine. Question 4.5 then. Doctors tested people of different ages to time how long it took between touching a sharp pin and the arm muscle contracting, and it shows your results. So you've got the increasing ages, okay, of the patients, and then you've got their time taken for their muscle to contract. So you can very much see the pattern quickly, what's happening. It says, describe the relationship between the age in years and the time taken for the muscle to respond and contract. Well, it's 2 marks, so it's a description question, and there's 2 marks, so we need to say 2 clear points. First of all, we can say the overall pattern, which is, as age increases, the time taken increases of the muscle to contract. We've got to think of a second point though, okay, to get our second mark. We've got the obvious one, but then we need to look a bit closer at the data. As it is increasing, it's not going up in a linear way. It's not going up an even amount each time. You can see the gap is increasing as the age of the tested people increases. So what we can say, it increases more quickly as age increases. So we're saying that if we were going to draw a graph of it, it would likely give a pattern like that. As your age increases, the time it takes for your muscles to respond goes up very quickly when you're older. Okay, so question 5 then. It says, read the following. In the 1950s, farmers in India could not grow enough rice to feed the increasing population. At the International Rice Research Institute, scientists began a selective breeding program with 10,000 varieties of rice plants. In 1966, the IRRI produced a new variety called IR8, which gave a yield up to 10 times the traditional varieties. IR8 has short stems and rice grains and large rice grains. IR8 was grown by farmers all over India, so people had enough to eat. It was produced by selective breeding. Describe the steps that scientists would have taken to produce IR8. So this is pretty much a 4-mark question saying describe how we do selective breeding, but relating it to IR8. So first stage of selective breeding is you select the features that you actually want to produce. So the farmers would have selected the features. Now in this situation, it's told us we want to have short stems and large like rice grains. Once we've selected our features, we would cross breed these samples. Okay, we would then collect the offspring and identify those with the desired characteristics again. We would then allow those ones only to breed again. So we would do this over multiple generations. Okay, continue to allow only those showing desired features to breed. So therefore, the desired genes are being passed on through the population and the less desired ones are not being produced. Okay, and you would keep on doing this until they all have the desired characteristics. Question 5.2. The IRRI has now developed several varieties of genetically modified rice plants. Some people in India agree and some people disagree with the GM varieties being grown. Explain why. So we can pretty much break this down into why do people agree with genetic modification and why do people disagree? Well, if we start off with agree, why do people like genetically modified plants? Well, quite simply, you have more food to feed the population. So that is good, and you would have a higher yield. You would then also have genetic modification can also give higher resistance to to pests, which gives a higher yield. So any little bugs trying to eat it, they would die themselves if they did eat the crop, so therefore the overall yield goes up. Okay, you can then also say that genetically modified crops may have a higher nutritional value so therefore people will be healthier and have a healthier population. So why might people be against GM plants? Well, we're not actually sure if you've got a really competitive, really successful plant, what it might do to wild plants. How might it affect ecosystems if it got out? It could affect habitats and population and food webs, etc. It may result in traditional varieties no longer being grown, being decreased in number, and as with all science and new science, it may affect long-term health, if not, as there could be not enough research conducted over the long term. Okay, I'm sure there would have been short-term research, but scientists may not have been able to do a 10-20 year study of the impact of eating these GM crops. Question 6 then. Figure 4 shows how hormone contraceptions, Okay, so question 6 then. Figure 4 shows how hormone concentrations vary during a normal human menstrual cycle if a woman does not become pregnant. You've got the different menstrual cycle hormones, FSH, LH, estrogen, progesterone. It says calculate the rate in increase in LH concentration between day 9 and day 12. Give your answer in arbitrary units per hour. So that's a key thing, per hour. Give it 2 significant figures. Well, we're looking at LH and we need to calculate its increase per hour between day 9 and day 12. So first thing we can do, we can go down identify which line is LH, which is a solid one. Day 9, we can see it is roughly at about, if I go across in arbitrary units, it's at about 17. So day 9 is 17. On day 12, it's at 50. Just reading off the graph there. So therefore, our change, how much it's gone up, okay, because we're calculating the rate of increase, well, it's gone up then by 33 arbitrary units. Now, it's asking us, not per day though, it's asking us per hour. So we've got a period of 1, 2, 3 days, okay, and we want it per hour. So 3 lots of 24 hours gives us 72. So we have had an increase of 33 arbitrary units over 72 hours. So we can then do 33 divided by 72, which gives us an answer of 0.45833. So it is going up by 0.45833 per hour across those 3 days on average, but it's after it by 2 significant figures, so I can draw my line there, and it's an 8 being the number before, so I round up 0.46 arbitrary units. Question 6.2. Describe the sequence of hormone interactions in the menstrual cycle. We've got to name where each hormone is produced. So this is effectively saying, describe the whole female menstrual cycle from the beginning, including where it is made and what those hormones do. So this is a really subject knowledge heavy question, and really does identify if you've revised it or not. So first of all then, the first hormone that is involved in the menstrual cycle is FSH. So number 1, FSH is made in the pituitary gland at the base of the brain. Okay, it travels in the blood to the ovaries. Its job is to mature the egg. It tells the ovaries to mature an egg. Possibly 2 if a bit too much FSH is produced. Then, once it hits the ovaries and it starts maturing an egg, the ovaries then produce estrogen, which is our second hormone. Estrogen has 2 jobs. Okay, so you could literally just say that. Estrogen has 2 roles. The first one, it stops FSH production because we don't want to mature all of the eggs in 1 month. We want to mature just a couple, or ideally just 1. And the second role is that it starts the lining of the uterus being developed. So it starts preparing that blood-rich lining. When estrogen then stops FSH, it then triggers the pituitary gland to start releasing LH. So when the pituitary stops FSH production, it starts LH. LH travels back to the ovaries, and its job is to tell the ovaries to release the egg. Okay, now the final hormone that is sometimes missed off is called progesterone. And although progesterone, its main job, you can just summarise it as been saying that it helps maintain the lining of the wall during pregnancy. Obviously, if pregnancy does not occur, then progesterone levels will fall, and that then causes menstruation, which is the shedding of the lining of the uterus. It then says, question 6.3, progesterone is used in some contraceptives. Suggest one advantage of using a progesterone patch rather than a progesterone oral contraceptive. So what's the advantage of using a patch rather than a pill? Okay, well, if we just think of a key advantage of why using a patch might be better, well, if you stick a patch on, it's going to last longer and therefore there's less chance of forgetting. So with a pill, there's a greater chance of forgetting to take it, or you can say a patch lasts longer. You could also say that the patch has a lower dose, so it would have fewer side effects, or the patch goes directly into the bloodstream, so it wouldn't be affected if that person was then sick at all. Question number 7 then. Many people eat shellfish called oysters. An oyster has 20 chromosomes in each body cell. Which arrangement of chromosomes will a male oyster have in each body cell? Now this is a classic situation of where they put standard biological knowledge in a really weird context. So although they're talking about oysters, it will relate to genetic inheritance and gender that you would learn in terms of humans. So what would a male starfish have? Well, if you remember that males in humans have X and Y chromosomes, so we need to look for that. Okay, so they would have one X and one Y chromosome and 9 pairs of others, okay, because they've got to then add up to 20. The other ones, so definitely can't have 2X chromosomes because that would be female. 2X and 2Y chromosomes and then 8 pairs, well that would give you too many chromosomes because you know they've only got 20. And if you had 1X and 1Y and 8 pairs of other chromosomes, that wouldn't give you enough. Oyster gametes only contain half the amount of DNA compared to normal oyster body cell. Describe the type of division that produces gametes. So just again, relate it back to humans. What do humans do to make their gametes? And it will be the same answer in terms of starfish. So the process of making gametes is called meiosis. And then we've got to describe meiosis. Stage 1 of meiosis is the DNA replicates. Then part number 2, once it's made and doubled its DNA, the cell splits once. Okay, there is at this stage DNA getting crossed over, so there's DNA variation. Then the cell will split again, making 4 cells with half the amount of genetic material, and these are called haploid cells. So a cell that has a full amount of DNA is called a diploid cell. These ones, if they have half the amount of DNA, are called haploid cell. And they are all, because there is variation, they are all genetically different. Biologists have discovered a way to produce oysters that have 3 sets of chromosomes, which are triploid, instead of the usual 2 sets that are diploid. The triploid oysters cannot reproduce, so they grow more quickly. Diploid oysters do not taste good in the reproductive season. Triploid oysters taste good all year. Figure 5 shows the chromosomes in a diploid cell and a triploid cell. Only 2 sets of chromosomes are shown. Suggest why the triploid oysters are not able to reproduce. Well, if they've got 3 sets of DNA instead of the normal 2, well, in meiosis, you've got cell splitting. So meiosis, you need to be able to half DNA. If you have 3 sets, this cannot be halved. (7.4) Explain why triploid oysters grow more quickly than the diploid oysters. So we need to find some information that we've been given about what may affect their growth rate. So it says that the triploid oysters cannot reproduce, so they grow more quickly. Why might that be? Well, if they're not reproducing, they're not then using energy reproducing. So less energy is used reproducing. So more energy is used to grow bigger. Therefore, they are growing bigger and they have more yield compared to the diploid that are spending their energy reproducing. 7.5 then. The population of diploid oysters growing in the world has reduced by over 80% in the last 20 years. Suggest 2 environmental factors that may be causing this reduction. Give a reason why each factor may be causing the reduction of the population. So the obvious factor that is probably reducing it is over-harvesting. But over-harvesting is not an environmental factor. That would be an economic factor. So we've got to just link it to environment. So what might be causing the amount of diploid oysters to go down? Well, it could just then be to do with global warming or climate change, because it may be increasing temperatures are killing the oysters. There could be other environmental factors, could just be pollution. Okay, maybe there's pollution from farmlands getting into the water. It may be increasing acidity, which is causing them to die or stop reproduction. You could also say that this is effectively thinking of reasons that are stopping or potentially even causing extinction. You could say there are new predators, or there could be new pathogens, or you could say new competitions or new competitors as well in the ecosystem. Question 7.6 then. Oyster farmers grow the triploid oysters from young seed oysters. The production of seed oysters involves the use of a chemical called cytochalazine B. Cytochalazine B has been shown to cause cancer in mice. Evaluate the production of triploid oysters for supermarkets and restaurants. So really, we are just evaluating, are these triploid oysters a good idea for supermarkets and restaurants? So what we can always do in this sort of question before you dive in and you write your answer is do a little comparison table. Okay, so you can have advantages, you can have disadvantages for using triploid oysters. So the advantages of using triploid oysters, as it mentioned previously in the question, they're good to eat all year. So always remember to look back in the question if there's any little hints that have been given. okay? And they are very popular. Because they are popular, then that is going to equal profit for the supermarket or restaurant. They also, as you've been told, grow faster because they're not reproducing. Therefore, there's going to be more supply. And as we know, the stocks can be replenished, so that is therefore more sustainable. Okay, so there's going to be a good amount of money to feed, and more oysters to feed a growing population. So what are the negatives of these triploid oysters then? Well, the big one that they put straight into the question just above it is that they can cause cancer in mice. Well, if they can cause cancer in mice, they might cause cancer in humans. Could it affect humans? It's not confirmed yet, but there's a chance it could be. So therefore, more research is probably needed. Another reason against it is that if you're using these manufactured triploid oysters, the diploid oysters could eventually become extinct. They're being outcompeted. Okay? And in terms of, because they cause cancer, well, that might put people off buying them. Okay? So less people buy. So that is a big economic disadvantage of using it. So although they would have loads of them, there might not be a big enough size in the population to then buy them. Okay? And then, last thing is that farmers would have to buy new seed oysters every time. Okay? So you would have to buy new seed oysters. That involves manufacturing them. So that could become quite expensive. So this paper was quite challenging. There are loads of different weird situations they've put the biological knowledge in. It's just a case of identifying whatever context they throw at you, they can only assess you with the biology that they've put in the specification. So just identify what part of the course it's talking about, and then you can try and link the context to it. So I've put the mark scheme and the blank paper on the right hand side. Please have a look. Yeah, best of luck. Thank you very much.